I want to talk for a couple minutes about chromatin structure and regulation in eukaryotic cells, because it's really interesting how we package our DNA and how that works with how much genes are expressed. So the functional unit of packaging DNA is called a nucleosome, and it's made when DNA associates with these proteins called histones. And there's a couple different varieties of histones. What's going to happen is they're going to form an octamer. So there's H2A, H2B, H3, H4. All those guys are going to be there twice, and those are going to make an octamer. And that is going to associate with DNA through charges. So these histones have a lot of the amino acid lysine, and lysine is positively charged because it's basic, and the basic amino group is going to grab a proton, and protons are positive, so it's going to be positive. And remember how DNA has a sugar-phosphate backbone? Well, phosphates are negative, so those two things are going to associate together. So the DNA and these histones are going to associate together because of the attraction of their charges. And they're going to form this unit called a nucleosome. And technically, a nucleosome is not just this. It's actually going to be that plus the linker DNA in between histones. So this is a nucleosome. And that's got about 200 nucleotides of DNA. So interesting fact, when your cells kill themselves, like if they're infected by something, or if their genetics get mutated too much, then the enzymes that cleave up the DNA cleave by the histone, and your DNA gets released in these 200 or so nucleotide bits. Anyways, so these histones are going to kind of roll up together and associate together, and this is where the histone H1 comes in, is it associates on these linker areas, and it helps all these histones come together, and they're going to come together into this really gigantic fiber. And this fiber is either going to hang out in the cell as chromatin, which is just when it's kind of like all squiggly and random, or when we're in the M phase, or the mitosis phase of the cell cycle, it's going to condense into a chromosome so that the genetic material can be easily divided. And these two parts of it are called sister chromatids. You can learn more about sister chromatids if you watch the videos on meiosis and mitosis. But anyways, the fun part comes when we think about how our genes are regulated through this. So when chromatin is really tightly wound up, we call that heterochromatin. So heterochromatin is going to be this really tightly wound up, inaccessible type of thing, because if your chromatin is really packed up like this, it's going to be really hard for DNA polymerase over here to get on in there and, RNA polymerase to get on in there and transcribe the DNA into RNA so it can get expressed as protein, right? So the other kind of chromatin that exists in our cells is called euchromatin, and euchromatin is a lot less compact and just a lot more relaxed. And this is going to allow for RNA polymerase to come on in and transcribe this DNA into RNA, and the RNA can go on to be protein. So how does this euchromatin or heterochromatin thing happen? Well, the association between DNA and histones depends on their attraction of charges, right? So there's actually these enzymes, they're called histone acetyltransferases, or HATs, and they will take one of the lysines, and lysine is an amino acid that ends with an amino group, and this amino group is going to be charged. In this case, it's going to be an NH3+. And these HATs, these histone acetyltransferases, are going to put an acyl group on there. So they're going to take away some of those hydrogens, and instead what's going to happen is that this is going to get acetylated, which looks like this. And now the charge is gone. And now the DNA says, I have a negative, but I can't find a positive, so I don't know why I'm supposed to stick around near this histone. So all of a sudden that association between the DNA and the histone breaks, and it's a lot easier for the DNA to be accessible to RNA polymerase and to be transcribed when that histone isn't in the way. There's also another enzyme called a histone deacetylase, and that can take away the acetyl group and put back that positive charge, reinstating the interaction between the histone and the DNA and making things bad for RNA polymerase to get in there. So the bottom line of this is that acetylation or other covalent modifications to histones, or to the lysine residues specifically in the histones, that's going to make genes be more transcribed. Whereas if you deacetylate histones, then your genes are going to be stuck in limbo and they're not going to be as transcribed. And this whole type of thing with modifications that don't necessarily change the gene, but they do change the levels of expression in the cell, we call this epigenetics. And these kind of things can actually be inherited, so like histone acetylation patterns and stuff can be inherited. Anyway, yeah, that's all I have to say about that, but pretty cool stuff, and pretty important too.